Hello everyone, my name is Suarez Castellanos and I'd like to thank you for the opportunity to come and present to you our work titled Design of Mixed Focus Ultrasound Patch Clamp Platform for the Study of Electrophysiological Activity, Neurostimulation or Neuromodulation by Focus Ultrasound. This is a work that was carried out at INSERM Lab 2 in Lyon, France. And the general objective of the study was to design and build a mixed hybrid platform that would integrate a focused ultrasound system with a technique in a platform that is commonly used in the field of neurosciences, which is the whole cell patch clamp technique. And this platform would allow us to study the electrophysiological activity that is generated by the application of focused ultrasound at the single cell level. And at the top of the poster, we can see a description of the experimental setup that we used. We have a picture on the left, as well as a schematic describing and showing the setup that was designed for this purpose. And to the right, we have a description of the cell models that were used for these studies. We used two cell models. The first one is primary neurons from mice, and these cell models were used for neuromodulation experiments. And we also used a human progenitor cell line REN cell that was differentiated into neurons, and these cells were used for neurostimulation experiments. So when we take a look at the results section immediately below the experimental setup section, we find the results that we obtained for both neuromodulation and neurostimulation experiments. In neuromodulation, we were interested in observing how the application of focused ultrasound to the targeted cell, the patch clamp cell, how the application of focused ultrasound would affect the activation threshold of these cells. And the activation threshold is defined as the amount of electrical energy required to stimulate the cell for triggering action potentials. And we found through our experiments that by applying focused ultrasound at very low pressures and very short pulses, that we were capable of both elevating and decreasing the activation threshold, therefore changing the amount of electrical energy that is required to stimulate the action potentials from these cells. Now, if we take a look at our neurostimulation experiments, we were interested in measuring the membrane potential of the patch cell in response to short pulses of focused ultrasound applied at very low pressures. And we were interested in seeing in detecting any sort of membrane depolarization or perturbation of the membrane potential as a result of this type of stimulation. And we varied two ultrasound parameters to highlight this effect. We varied both the pressure applied and the pulse duration. On the top graphs, we can see that by progressively increasing the pressure of the focused ultrasound stimulation from 6 kilopascals to 25 kilopascals, we were able to detect different kinds of either immediate or delayed membrane depolarizations from the patch cell. Similarly, when we progressively increased the pulse duration in the lower graph, when we increased the pulse duration from 712 cycles to 11,000 cycles, we were also able to detect delayed membrane depolarizations as a result of this treatment. So in conclusion, we have demonstrated that a mixed platform that integrates a focused ultrasound stimulation system and a whole cell patch clamp platform is useful for the study of the electrophysiological activity produced, modulated and stimulated by focused ultrasound, and could be a valuable tool for the study of the biomechanisms involved in this process. I would like to thank our funding sources, the ANR in France, the Lab Extend We Can, and the Focus Ultrasound Foundation. Thank you for your time, and I will now take questions from the audience.